Good morning, everyone. Welcome, everyone. Let me add my welcome to Amy's welcome. As she said, we have almost 750 people registered for this meeting, which is about threefold more than we had last year, which was about 2.5-fold more than we had the first year. And so I think the growth in interest in this meeting, the growth in the audience for our podcast series, and the increasing readership of Lessons in Antisense are evidences that we're making progress toward two of our really important goals. The first is to create a community for the Nano-rare patient and family, a knowledgeable community. And second, to draw attention to the needs of Nano-rare patients and families. And I think as we do that, we begin to see movement toward more holistic long-term solutions. Our task is here today to help the patients we can. And I believe that in doing that, we create the best momentum to drive even more functional solutions long-term. And so we're very excited about the opportunity to share with you all that we're learning in the next couple of days, and excited to see so many people here, and to welcome so many new members of the n-Lorem family. We exist for the patient and the family. And so when you join us, it makes a big difference. So I have noticed that many of the rare disease organizations divide their meetings into patient and family sessions and scientific sessions. And we've chosen to make our meeting fully integrated. And we've done that for a number of important reasons. First, we think patients and families must be full partners in this effort. And we will invite you to be partners with us in this endeavor. And then maybe even more importantly, patients and families are called upon to make very difficult decisions. And I think you can do that better if you have a basic understanding of the strengths and limits of ASO technology, the advances that we're making in the technology, and you get to see firsthand, unfiltered by any expert, the kind of information we're delivering, the kind of data we're seeing, the kind of results that we're getting. And so our plan is to continue the meeting as a fully integrated meeting. We think that is the best solution, but we are certainly all ears. So if you have suggestions about how we might make the meeting better, just jot Amy or me a note, and we'll certainly consider all those suggestions. You know, the demand has, just extraordinary, and it's just exceeded any demand that I could have imagined. And that's meant that we've had to raise a lot more money a lot faster than I ever thought possible, and to grow much faster than certainly was planned. And that's really challenged us. And even with all the money we're raising, there's still not enough money to treat everyone as fast as we'd like to treat them. But I am proud of the fact that if you ask who's treated nano-rare patients today with ASOs, 95% of the patients treated have been treated by n-Lorem. And we've treated our patients safely and effectively. And I think that's important. It's also meant that we've learned a lot more than we would have learned if we had just had the demand that we expected. And so our goal in this meeting is to share what we've learned with you. And we think we've learned a lot. And of course, it teaches us about the genes, the mutations, the phenotypes, and the responses of nano-rare patients. But the lessons that we're learning, because nano-rare patients are unique experiments of nature in whom we have a single variable introduced driving enormous phenotypic changes. And from you, we're learning lessons that I believe will, in due course, change the way we think about health and disease altogether. And it's high time we do that. So those are the lessons we want to share with you in the next 2 days. And it will be a busy 2 days. But before we do that, I want to take a minute to put what we're doing here today and tomorrow into a broader context. When you make your way to one of the outstanding physician investigators who take care of patients and collaborate with us, the first couple of things they're likely to say to you is, we need a little sample so we can sequence your genes. And we also need to just take a little biopsy of your skin so we can get some of your cells, and we can study your disease in your cells. Said that way, it seems routine, even pedestrian. But the investigator could have said, thanks to several miracles of science, today, for the first time in history, I can take a small sample from you, decode your entire genetic library, pinpoint the cause of your disease, and in doing that, facilitate the creation of a genetic medicine directed to the cause of your disease to be created just for you. Said that way, it doesn't seem pedestrian at all. Your investigator could have said, thanks to some other miracles of science, I can take a little sample of your skin, collect some cells, we'll call them fibroblasts, treat them in a certain way, cause them to change into cells of interest to study for you, and then evaluate your disease in your own cells, develop a medicine for you in your own cells. And we're learning that if we have an ASO that addresses all the problems of your cells, it's likely will address all your problems. Said that way, it seems like a very different proposition, doesn't it? And that's what we're doing. And I do believe that what we're doing is miraculous. And so I want to make sure that you understand and take every now and then, reflect on the fact that what we're doing is truly astonishing. And it all begins with curiosity. We're curious by nature, and there have always been humans, some humans in a group of humans, who are more curious than others. And over time, those humans learned that if they practiced a more organized kind of curiosity, they learned more efficiently. They learned more rapidly. And they needed a name to call those people, so we call them scientists. And the work that they do is science. But in fact, it's just the organized practice of curiosity. Nothing more, nothing less. And it's a product of our intrinsic curiosity. And so everything begins with a question, curiosity, and an organized approach to answering those questions, science. And I'm quite certain that never in a presentation I've ever given, and I've given a lot, have I used the word miracle. it's not a word that I even think of using. But the events that I'm going to be describing in the next few minutes changed the world, changed the way we understand ourselves, the way we understand health and disease, changed us, changed the present, and continue to change the future. And that sounds pretty miraculous to me. But they're not like fairy tale miracles. We don't get some lamp that we can rub and a genie pop out and give us 3 wishes. I'd ask for 10. I don't know why nobody asked for 10. I'd ask for 10. Probably bought me for being greedy, I guess. But in any event, we don't get to do that. The miracles I'm talking about are created by human beings, and they affect all of humankind. And they take decades, often centuries, to create. So those are the miracles that we want to talk about in the next few minutes before we begin to focus on the details of what we're learning. And so let's talk about the miracles of genomics. I think most people know that the idea of sequencing the human genome began in 1900. It didn't. It began when the first human realized that information was transferred from parent to progeny. And so that's been known by humans since humans were around. And as soon as we became farmers, instead of hunters and gatherers, what did we do? We began to breed plants and animals. And so we've known that there had to be some kind of genetic information forever. But it wasn't until 1866 when the rules of transfer of genetic information were understood by Mendel. And so in 1866, this is now more than 100 years before the genomic revolution, we understood what genetic information, the rules of genetic information transfer. But that left the billion-dollar questions, didn't it? How could a biochemical encode, use, retrieve information? How could that be done? What is the structure? What is the chemistry of a gene? Unknown. And so that led to basically 100 years of work. And there were many candidate molecules, lots of controversy because science is controversial. And in fact, DNA was sort of an also-ran. It was considered, just another uninteresting carbohydrate like starch for many years. And often what happens in science is that there's an invention in an entirely unrelated field that changes a lot. And that was the case with the invention of X-ray crystallography. X-ray crystallography was invented to understand the structure of crystals. And when that technique came available, people started crystallizing proteins and other things to understand their structure. And in 1954, Watson and Crick crystallized DNA and intuited from the crystal structure the genetic alphabet, the nucleotides A, T, C, G, and how genetic information is encoded, stored, and used. And that breakthrough changed the world. In my view, it changed the world as much as E equals MC squared, as much as nuclear power. It made everything that we do today possible. And it continues to influence our future in ways that I find truly astonishing. And so, after DNA was crystallized, decades of work, and that then led to the Human Genome Project that was begun in 1990, completed in 2003. It is a revolution, but it started centuries ago, punctuated by Watson and Crick in 1954, and still delivering incredible value today. That is the miracle of genomics. Without that, there would be no n-Lorem, there would be no ASOs, there would be no hope, there would be no help, there would be no benefit. Centuries in the making. And then let's now talk about the miracles of iPSCs. Well, first of all, what in the world is iPSCs? Hang on a minute, I'm going to tell you. About half the time I say it wrong. I say ISPCs and people correct me. They're very smug when they correct me. But anyway, that's just my problem. But we'll come to what an iPSC is. So, of course, we've always been curious. Begins with curiosity. Everything begins with curiosity. And we've always wondered how we work, what we're made of, and so on. And why do we get sick? Why do we fail? In 1590, a father and son team of opticians named the Janssens reinvented the microscope. Actually, the microscope's been invented many times in history. Thousands of years before the Janssens, the Chinese had a microscope where they used different columns of water to change the magnification. I can't imagine that working, and they must have had awfully pure water because it sure would have been murky in some of the places I've looked, but in any event, they had it. But in 1590, the microscope was reinvented, and we realized that we're made up of trillions of these tiny little units, needed a name to call them, we called them cells. And other scientists noticed that we're not made up of just one kind of cell, we have all different kinds of cells. And those two observations led to centuries of work, centuries. First, to answer the question, what is a cell? How does it work? What does it do? Why do cells get sick? And that, of course, we called molecular and cellular biology. That is the molecular basis of how cells function. Hundreds of years of work. And then others were interested in how we start as a single cell and become so many different kinds of cells. Needed a name to call that, so it was called differentiation. And so these two themes of science traveled along for centuries and came together in 2006, just 20 years ago, just a blink in time. And of course, that was the discovery of iPSCs by Yamanaka and the labs and his colleagues in Japan. And what was realized by many is that even when you have mature organs, you have cells die, and they have to be replaced, and it seems that they're replaced by resident cells in the organ. And those resident cells in the organ then are stem cells. That is, they retain the ability to divide and change. And a lot of them look like fibroblasts. And so we'll call them fibroblasts for the moment. These fibroblasts could be, Yamanaka and colleagues said, why don't we take those fibroblasts, put them in the lab, treat them with some biochemicals that we know cause differentiation in utero, and see if we can change them into cells of interest that we could use to study. iPSCs, induced pluripotent stem cells. They're stem cells because they can divide. They're pluripotent because they're not omnipotent. They can't be anything, so they can be some things, pluri. And they're induced because we treat them with biochemicals to cause them to change into the cells that we want to study. And we use those cells every day in the lab to create the medicines that we bring to you. And we are learning the amazing fact that if we correct the entire cellular phenotype, it's likely we'll correct the entire phenotype of the patient. Just that is a truly amazing observation. So, centuries of work. and that induced pluripotent stem cells. So now you know iPSC. So one more miracle that I find really kind of wonderful, and that's the miracle of the RNA world. You know, shortly after DNA was discovered, it was discovered there was another molecule that looked just like DNA, but it had a sugar that was a little different. It's ribose, not deoxyribose, so it's called RNA. But unlike DNA, it wasn't concentrated in the center of the cell. It seemed to be spread throughout the cell. And the more we looked, the more kinds of RNA there were. And there were big controversies, big wars. In fact, when I started as a graduate student, we were trying to sequence what's called U-rich RNA, and there were still lots of people who thought they were degradation products. And so there were big fights in science. Are these RNAs real? And it turns out that they're all real. They all exist for a reason. They all have different functions, and they come in different sizes and different shapes. And over the years, we've come to understand the RNA world. And this is the world that I've lived in now since I was a graduate student. And I wish I could describe it for you in the way I see it, because it's a marvelous world. It's just a beautiful world. And so the third miracle is the RNA world. And it's another source of what we do. And in 1978, and I should find a different name because it's not a breakthrough. This was just an idea. Certainly not a breakthrough in the way the other breakthroughs that I talked about happen. But in 1978, Zamasek and Stevenson realized that RNA is single-stranded, which means that you could use the genetic code itself to design a drug that would bind to a target RNA using genetic information. And since it would be comprised of a few nucleotides, they said, let's call it oligonucleotides. And since genetic information makes sense when read left to right, and this oligonucleotide would be right to left, we'll call it the antisense strand. Antisense oligonucleotides, our technology. And that percolated for a while, and in 1987-89, there were several companies formed to see if this platform for drug discovery could be created. Among them was Ionis, the company I founded. And the history of antisense technology is odd compared to, say, monoclonal antibodies, gene therapy, even CRISPR, in that there were a few companies that got founded, but almost all the companies gave up, failed, changed directions, except for Ionis. And there was really no real interest in academia about this technology. So for the better part of 25 years, one little company had to go it alone. That was Ionis. So would I call antisense a miracle? No, probably not. But I will tell you that there were days when I thought Ionis was going to fail. So in that sense, it was very miraculous to me that we're still around and we're still here. And that led to the creation of n-Lorem in 2020. So this sweep of science that is centuries in the making bring us together today. That's an amazing story that I want you to reflect on as you begin to focus on the lessons that we're going to be sharing and the details of what we're learning. And that this story demonstrates another very important principle. Knowledge is power. As knowledge accumulates, power multiplies. And it multiplies because we, today, operate from a scaffold of knowledge that's been constructed over centuries. We stand on the shoulders of the great scientists and physicians, generations of them that preceded us. And it is fragile. It is fragile. And today, in ways that I never dreamt I'd see, that the pursuit of knowledge through science is under assault. Everyone in this audience is a direct beneficiary of the pursuit of knowledge by science. So you can't be neutral. You can't stand by and let that happen. And you don't have to ask what happens if you disrupt knowledge and the pursuit of knowledge. We know it's already been tried. It was centuries of ignorance and squalor that are aptly called the Dark Ages. There is that risk today. And I put it to you, you can't be passive. We cannot allow knowledge and the pursuit of knowledge through science to be lost. Everybody with me? You really need to know that. So, over the next couple of days, I want you to focus on the details, of course. But just every now and then, step back for a minute and think about what a miracle it is that I can talk about doing the things that we talk about as routine today. And I believe that our pioneer Nano-rare patients and the work we're doing will be a part of the leading edge of science that drives one of the next revolutions in medicine. And I think it's long past due. I look to a day in the near future when we discard the traditional names of diseases. They're just archaic descriptions of what people look like when they get sick enough to go to the doctor. Most of them are centuries old, and they mean absolutely nothing from a molecular level. So it's time to get rid of them. They just get in the way. Instead, we focus on what is a healthy phenotype. You know that if you look up health in the dictionary, one definition you find is the absence of disease. Something as important as health defined as the absence of something bad. And the other definitions are equally vague and dumb. We don't know what a healthy phenotype is. We do know that there are healthy phenotypes, and we understand vaguely how healthy phenotypes are maintained. And so I think it's time now to focus on understanding what a healthy phenotype is, understand how we maintain a healthy phenotype, and then if we're forced to think about disease, we ask, what is the cause? And we address the cause, not the symptoms. And if you ask, what do we do every day at n-Lorem? We ask, what is the cause? We don't care about the name of the disease. We ask, what is the cause? We make a medicine to address the cause of the disease, and then we treat the cause of the disease. That is, I think, the future of medicine. And if we then encourage the introduction of sequencing of all newborns, which is happening in other parts of the world, but not in the US, then we'll know the humans at risk of having a cause of a disease, and we'll be able to intervene much earlier in diseases. And that will be a very fundamental change. We certainly know that if we treat earlier, the outcomes are better. That's across the board in medicine. And that stands today as a possibility, as a real possibility for the first time in history. So, focus on the details. We look forward to sharing all that we're learning with you and all the challenges that we face as well. But remember what a miracle it is that we can do what we're doing and how much we owe generation after generation of science and the pursuit of knowledge through science for being here today. Thanks, everybody.